Good morning, everybody. I'll be talking about the physiology and spatial specificity of functional MRI. When we introduce a subject into the magnet, present sensory stimuli or motor tasks, we measure T2 star or T2 or cerebral blood flow signals. In between, there are changes in neuronal activity and changes in the vasculature. Although we measure functional MRI, most of us are really interested in changes in neuronal activity. We measure functional MRI because this is, of all non-invasive imaging methods, this is the method with the highest spatial resolution, spatial specificity as well. So in order to understand what happens in terms of neuronal activity, we need to learn about the transformation between neuronal activity and functional MRI and the inverse transformation. So I'll talk a little bit about the basics of intracranial neurophysiology, about the physiological mechanism of the bold response, the negative bold response, resting state bold, and the spatial specificity of bold response relative to neurophysiological and metabolic activity. We'll start by introducing basic concepts of neurophysiological recordings. Neurons in the model of extracellular recordings are considered to be embedded in an extracellular medium that acts as a volume conductor. For all of us that measure local field potential or EEG or MEG, it is important to note that cortex and the brain in general is a volume conductor. When 2 points within a neuron have different potential, there is a flow of primary current, the blue one here. Unfortunately, the mouse doesn't work for me to point at, but you can see the blue arrow. That's a primary current within the neuron, and there is a nullifying current, a secondary current, through the extracellular matrix, because the extracellular matrix is conductive. So we measure exactly this extracellular nullifying current. Here is an example of recordings from one electrode. We can filter this signal to frequencies that are higher than 400 hertz and to frequencies that are lower than 150 hertz. We consider the higher frequencies to represent multi-unit activity or spiking activity, and the lower frequencies to represent synchronized synaptic activity. Why do we consider it this way? Because if we look at spectra of action potentials, we can see that the energy is mainly focused at 400, 500 hertz and above. And in contrast, if we look at spectra of EPSPs, excitatory postsynaptic potentials, we can see that it is focused mainly with power below 100 or 150 hertz. So local field potential that represents synaptic activity and multi-unit activity can be separated in the frequency domain, and obviously local field potentials can be further classified to frequency bands that are used classically in the EEG literature. What constitutes the multi-unit activity and the local field potential? Multi-unit activity, as we said, we believe that it represents action potentials, very short events that are with origin very close to the electrode, on the order of 100 micron from the electrode. Local field potential, single event duration, can be 10 to 100 millisecond, and it can record data or postsynaptic potentials from up to 1 or 2 millimeter away because cortex is conductive. It represents population synaptic potential and the input to a given cortical area, but importantly, also the local intracortical processing, because the majority of synapses on any neuron within cortex are with origin of 200 microns away as a limit, the limit of the origin of the distance from that neuron. So it represents also local intracortical processing. Before we move on to functional MRI and to the vasculature, here is a transition slide that actually tells us that functional MRI signal is an indicator of overall activity of very many neurons and processes, because within a conventional voxel at 3 Tesla, say 2 by 2 by 2 millimeters, there are approximately 1 million neurons and 10 to the 10th synapses. So we move on now, and we talk about the physiological mechanism of the BOLD response. How do changes in the vasculature end up as measurement of T2 star signal? The BOLD principle is based on the fact that the oxyhemoglobin is paramagnetic. It has some weak properties, magnetic properties, as opposed to oxyhemoglobin, which is diamagnetic. It is very, very, very weak. When the oxyhemoglobin molecules are introduced within otherwise homogeneous magnetic field, they change the flux of the magnetic field and make it non-homogeneous. We measure functional MRI in specific areas. This means that there is some control of where blood flow goes to. So it's not like just increase in blood flow throughout the entire brain. We believe that the control is by arterial smooth muscles around the arteries and by pericytes that give rise to processes around capillaries and are capable of making the capillaries narrow or dilated. Most of us measure the positive BOLD response, and the sequence of events in the positive BOLD response is as follows. There is a change in neuronal activity, increase in neuronal activity that causes increase in oxygen consumption. This increase in oxygen consumption, or actually it's already the change in neuronal activity in a feed-forward manner, causes quite quickly to dilation of the arteries and increase in blood flow in an overcompensation manner. You can see that I wrote over there, I indicated 2 arrows of increase in blood flow relative to only 1 arrow in increase in oxygen consumption. So there is overcompensation that increases the oxyhemoglobin and washes out the oxyhemoglobin. Therefore, the magnetic field becomes more homogeneous, the T2 star signal increases, and the BOLD signal, or the T2 star signal increases. That's the positive BOLD response. We want now to relate metabolic, hemodynamic, and functional MRI signals to neuronal activity in a more direct manner. So I'll start by introducing a system of optical imaging which measures blood oxygenation and blood volume. The physiology of the hemodynamic response here is identical to that we measure with BOLD functional MRI. The only difference is that instead of looking at magnetic properties of hemoglobin, we look at absorption spectra of hemoglobin, or more specifically, differences between absorption of the oxyhemoglobin in blue in the plot over there, and oxyhemoglobin in red. You can see that at 616 nanometer illumination, the oxyhemoglobin absorbs more than oxyhemoglobin. And we'll combine it with measurement of voltage-sensitive dyes. These are compounds that we apply topically to cortex. The compound binds to the membrane of the neurons, and it fluoresces in quite a linear manner with the membrane potential. We use 2 cameras, and we can measure both the voltage-sensitive dye and the intrinsic signal. And I want you to pay attention to the time lag between the neurophysiological response and the hemodynamic response, origin of BOLD signal, gray matter or vessels, where do we get more blood oxygenation, and the spatial specificity relative to neuronal activity. So on the left, we have the imaging of the membrane potential with voltage-sensitive dye. At the bottom is the time course. This is 4-pore stimulation, electrical stimulation, to the 4-pore of the rat. And on the right, we see from the same region the blood oxygenation signal and the time course. Is it possible to start the movie, please? Technicians, is it possible to start the movies? So right now, there was a flash. That was the response to a FOPO stimulation. It's gone a long time ago. Now the hemodynamic response develops. Is it possible to do it again, please? Doesn't look right. Flash, development of the hemodynamic response, mainly in arteries, just a little bit in gray matter at a lag. We want now to move on, and just from demonstration to look at quantitative relationship. And here is a classical paper from 2000 from the lab of David Heger, who presented to primates and to humans visual stimuli at different luminance contrast. You can see at the left, this is low contrast. On the right, a high contrast. The low goes very close to 0, the high up to about 80% contrast. And it is known from monkey physiology that the higher the contrast, the luminance contrast, the higher the response of neurons in area V1. This can be seen by the single neurons, the red curve that was recorded from primates in area V1. The dark circles represent measurement of functional MRI in human subjects, also in V1 with the same visual stimulus. You can see that although the two responses are non-linear with luminance contrast, the curves obtained from the two measurements pretty much follow each other, suggesting that positive ball responses in human V1 are proportional to average firing rates in monkey V1. Similarly, if we look at the paper, a classical paper from the Lauritsen group in the cerebellum, stimulation of the climbing fibers and the parallel fibers during neurophysiological recordings from Purkinje cells and laser Doppler, we can see that a stimulation of the climbing fibers causes linear increase in cerebellar blood flow as a function of local field potential. And on the right, stimulation of the parallel fibers causes monotonous increase, but it's more of a sigmoidal function relative to the local field potential. So the hemodynamic response in many instances is approximately linear with the underlying neuronal spike activity. Non-linearities between the hemodynamic response and neuronal activity have been observed. In the vast majority of cases, the hemodynamic response and the bold response increase monotonously with increasing neurophysiological activity. Which type of neuronal activity is reflected by bold, spikes or synaptic activity? We already showed that positive bold response in human V1 are proportional to average firing rates in V1 of the monkey cortex. So does the bold reflect spiking activity? Obviously, not necessarily, because correlation does not mean causation. And it is possible that action potential, spiking activity, is correlated with bold response through a third mechanism, such as synaptic activity, that we can measure with local field potential. And indeed, from the same classical paper from Lauritsen, we can see here at the bottom stimulation of the parallel fibers. That's where you see the blips at the bottom curve that causes increases in cerebellar blood flow, although in the top plot, the number of action potentials diminishes. There's almost no action potentials of the local Purkinje cells. So this demonstrates a dissociation of spike rate and blood flow, which means that cerebellar blood flow does not depend on action potential and is likely to reflect synaptic activity. In cortex, things are more complicated because everything is connected to everything locally. So here's a demonstration from another classical paper by Logothetis et al. The black curve represents the extracellular recordings. The yellow curve represents the decimated or the overall envelope of the extracellular recordings, and the red curve showing the lag in a very clear manner, the bold response associated with this increase in neurophysiological activity. So in this slide, we see here, again, a response to rotating checkers in the visual cortex. The electrode is in V1, and the duration of the stimulus changes from 24 seconds on the upper panel to 12 seconds in the middle panel and 4 seconds at the bottom panel. We can see in the background, in pink, the bold response, and we can see that the bold response follows the duration of the stimulus with the lag. In contrast, the spike density function or the multi-unit activity in green and in blue are transient. They show only a transient response, so they cannot really explain or account for these long, bold responses. In contrast, if we look at the local field potential in the gamma domain, it is prolonged similarly to the stimulus and can explain better, because of that, can explain better the bold response. So the bold response reflects changes in local field potential or synaptic activity, input to and local processing in a region more than multi-unit activity or output of a region. I'll devote 2 slides here to a very important topic about signal-to-noise ratio, or better phrased, contrast-to-noise ratio of neurophysiological responses and functional MRI. From the same study of logothetis, we can see a signal-to-noise ratio for neural and homodynamic signals. The stimulus, the visual stimulus, starts at 10 seconds on the time, on the horizontal axis of the time. To the left, we see the neuronal activity in standard deviation units of the baseline, during the baseline before the onset of the stimulus. And the neuronal activity in standard deviation units is represented by the greenish surface. In contrast, the blue curve represents the bold response. And again, in standard deviation units of the bold baseline before the onset of the stimulus, and the scale is represented to the right. So we can see on the left that neuronal response, the onset is almost immediate upon presentation of the stimulus, and it gets to 60 standard deviations higher than the baseline of the neurophysiological fluctuations before the presentation of the stimulus. In contrast, after 6 seconds from the onset of the stimulation, the bold response is at about 2.2 standard deviations above its own, or relative to its own baseline. This means that the contrast-to-noise ratio of neurophysiology, neurophysiological recordings, is higher, much higher than that of the bold signal, and therefore, thresholding methods and not sufficiently averaging are likely to underestimate a great deal of actual neuronal activity related to the stimulus or task. It was actually shown later on by the Bandettini group that if you take a subject and you repeat a paradigm of functional MRI over 5 sessions, you can increase, or you get many more additional areas that show responses. Virtually the entire brain responds to a sensory stimulus or motor task. Many areas are involved, and the only way to get to that is by averaging a lot, much more than what we are used to. So I'll change gear now, and I'll talk about negative bold responses that we can see here in response to partial visual field stimulation, stimulation of only the visual field with flickering checkers that shows positive bold response, or creates positive bold response in the areas where the checkers are represented retinotopically, and negative bold responses otherwise. Here it is shown both in bold to the left and cerebral blood flow to the right in the human visual cortex. Now, mechanism could be interpreted as either vascular, meaning increases in cerebral blood flow to one area causes, through vascular effects, reduced cerebral blood flow in another area, or it could be interpreted as a neuronal effect, meaning increases in neuronal activity in one area induces suppression of neuronal activity in another that causes decrease in cerebral blood flow. And of course, choosing or selecting the right one per your paradigm is extremely important. In one case, we can interpret the negative ball response as an artifact. In another case, as information which is valuable response to the stimulus or to the task. So that's a study that I pursued together with Nikos Logothetis. We can see an electrode near the green arrow in V1 of monkeys. And to the left, we can see the receptive field of the neurons near the electrode in green. And we used two stimuli. One stimulus overlapped the receptive field, and the other did not overlap the receptive field. As we expected already from the human study, the overlapping stimulus induced positive ball response near the electrode. The non-overlapping stimulus induced negative ball response. Here's we use the region of interest around the electrode in the gray matter, sampled the ball response, and indeed we see an average of several sessions here, a negative ball response with the non-overlapping stimulus, not also the sort of in the blue curve of the negative ball response. When the stimulus is released, you can see an overshoot in the negative ball response that can also be seen in the neurophysiological data that was recorded simultaneously. Immediately at the end of the stimulus, you can see an overshoot. And of course, during the stimulus, you can see decreases in neuronal activity associated with the negative ball response. So that's the mean power of the local field potential. And this phenomenon can be also seen in just measuring action potentials. And therefore, negative ball responses in this paradigm can be interpreted as decreases or cerebral blood flow decrease due to decrease in neuronal activity. A recent paper used optogenetics in order to study the mechanism of negative ball responses. So Ulrova from Anna Davor's lab used optogenetics in order to selectively activate inhibitory neurons in the somatosensory cortex of mice, and she showed an increase in hemodynamic, or in this case, in dilation of arteries, arterioles and arteries, in response to optogenetic stimulation of inhibitory neurons that is followed by a prolonged constriction of the arteries, and they suggest, or these results suggest, that inhibitory neurons may be a possible cause for negative ball responses. Conclusions regarding negative ball response: in a majority of study cases, negative ball responses are associated with decreases in neurophysiological activity. Negative ball responses that are not spatially adjacent to positive ball responses most likely reflect decreases in neuronal activity. We'll talk now about neuronal activity underlying resting state bold. We all know that many studies have been conducted about resting state spontaneous activity and functional connectivity based on functional MRI, spontaneous fluctuations. Do these spontaneous fluctuations reflect neurophysiological activity? This is not trivial that they do reflect neurophysiological activity, because several papers, including the one I present here from Beer et al., showed that physiological fluctuations, subject motion, et cetera, do contribute significantly to spontaneous fluctuations in fMRI signal. In this case, we see that the respiration volume per time correlates with functional MRI signal and creates a map that can be interpreted as a network during resting state. So we need to be careful about that. In a study that I conducted with David Leopold, We measured simultaneously functional MRI in monkey V1 together with neurophysiological recording simultaneously. In order to get the two signals to the same time domain of 1 sample per second, we computed the spectrogram that you can see on the top right from the neurophysiological signal, and we sampled it at 1 second, which was identical to the TR of the functional MRI. So now we can correlate power of gamma or multi-unit activity, or just measuring the number of action potentials per time with the fluctuations in bolt signal. When we do that, to the left, we can see a hemodynamic response-like function of the correlation between fluctuations in bolt and neurophysiological activity that on the right is gone if we break the simultaneity condition, meaning if we shuffle the neurophysiological recordings relative to the fluctuations in bolt, suggesting that this is not a false positive result. So this is in the broadband and in gamma activity, multi-unit activity, and spike rate. The spatial extent of the correlation relative to the site of where we recorded with neurophysiology is quite wide in the visual cortex, pretty much whatever the surface call of functional MRI was able to detect. Here is a follow-up from David Leopold's study, again showing that the spatial extent is actually even more than just the visual cortex. It goes to parietal cortex as well, which confirms what we know, or is in line with what we know from resting state in humans about long-range functional correlations in the resting state. So spontaneous fluctuations in fMRI signals in V1 correlate with the locally measured fluctuations in the local field potential gamma activity, multi-unit activity, and spike rate. The neuronal and hemodynamic signals are correlated over large regions of the visual cortex. I want to add that recent studies showed that independent of these results, on top of these results, there are likely also very slow fluctuations in membrane potential, in fluctuations that are as slow as resting state fluctuations in functional MRI, and both contribute, both the results that I present here and the very slow fluctuations contribute to fluctuations in fMRI signal. So are there cases in which a clear neuronal cause to changes in bolt signal cannot be found? So here is one example. There are not that many, but here is one example from Das laboratory in the monkey. We can see to the left, where stimulus is written in red, we can see responses of blood volume. They are inverted because that's optical imaging. We can see responses to visual stimuli. And on the right, we see weaker responses in dark. And these responses, as shown by the Das laboratory, are there simply because of the progression of the trial-to-trial expectation by the monkey. And they have no, as far as Das measured, they have no clear neurophysiological correlates. Possible explanations is that this is an effect of neuromodulators, including noradrenaline, which is released by the locus coeruleus, when the primate has a task and he expects to apply or pursue this task. So the noradrenaline level increases in conjunction with the trial structure. Conclusion, neuronal correlates of functional MRI bolt signals under most of the conditions, but not always, but most of the conditions, functional MRI signals are approximately proportional to the overall local neuronal activity. I believe that to draw more conditions or more conclusions when using just non-invasive functional MRI is difficult. But this take-home message, I think, is very strong for the very, very majority of functional MRI measurements. Changes in functional MRI reflect changes in the overall neuronal activity within the voxel. So with the permission of Tor and Nico, I will use 3 more minutes to present the last topic, which is the spatial specificity of the BOLD response relative to neurophysiological and metabolic activity. Here is a recent study from Vanderbilt University, the lab of John Gore, that imaged in marmosets, a small primate, BOLD, and to the left, and followed by neurophysiological recordings from arrays on the right. So it's not simultaneous, it's in succession. And they use the same stimulation of digits and measured responses in the somatosensory region of the marmoset. And what they show is that the profile of the response to BOLD and neurophysiological activity is very similar. And if they measure at the bottom left the full width at half max of the point spread function, they get both for BOLD and local field potential about 1 millimeter, assuming that point spread function is a Gaussian. We obtained recently similar results in humans. The principle is that we observed that previous studies that estimated the spatial specificity, including my own study, used retinotopic stimuli in the visual cortex, partial visual field stimulation. However, this stimulus is known from monkey physiology to create scatter of neuronal response. The neuronal response itself has quite a certain and reasonable spread. So previous estimation of the point spread function in humans based on BOLD response account for both the spread of the neurophysiological activity and the BOLD response. In order to circumvent this problem and to measure or estimate the point spread function of the BOLD response itself, we looked at point spread function of differential spin echo BOLD and gradient echo BOLD responses in humans at 7 Tesla while avoiding neurophysiological spread confound. We did that by fitting a model of imaging cortical columns presented previously by Chaimov et al. In 2011. We fitted this model to measurement of ocular dominance that Issa Yacoub and I performed previously at the University of Minnesota. So, and we also obtained, we constrained the parameter space based on post-mortem cytochrome oxidase histology from humans from Adams et al. So the idea is the following. The model to the left creates it in each iteration a probable neurophysiological activity map of ocular dominance response. It then simulates the BOLD response to the bottom left, and it checks what is the probability of getting this simulated BOLD response given the current parameters and the data which is presented to the right. What you see here is a demonstration of the first iterations, about 1% first iterations of this simulation and fitting of the data to our, fitting of the model to the data. Is it possible to run the movie, please? So you can see at the bottom the simulated ocular dominance. At the top, the measured ocular dominance. Pretty soon, the simulated, or pretty quickly after the onset of the simulation, the ocular dominance, the simulated ocular dominance converge to the actual measured ocular dominance. And on the right, you can see the width of the point spread function of spin echo, which converges to about 0.8 millimeter, and gradient echo to about 1 millimeter. So our results are similar to what John Gore obtained in the primate, and this takes in account already the spread of the neuronal response. That's the final result. We get the probability density function for the point spread of spin echo responses and gradient echo responses that are about, for gradient echo, 1 millimeter, and spin echo, 0.8 millimeter. So conclusions regarding the spatial specificity of the BOLD response. The BOLD response, especially under conditions of differential analysis, one stimulus minus the other, reflects the underlying neuronal responses in high fidelity. I would like to thank you for your attention.